is Kirsten Brandt, and I'm a senior lecturer here at Newcastle University. And I'll talk with you about carrots and cancer. You should be able to see the slide. So I'll talk about something about what we know about this, about carrots and cancer, some of the things we don't know. And I'll also try to explain a bit why it takes so long to find out, because you'd think that by now we would have the perfect pill made out of the carrots, and we still don't. And we probably will not have that sort of any time coming up soon. So if I can make this screen work, yes, it now works. So everybody knows, because you've been told all the time about vegetables, that they are really good for your health and very nutritious. And you should eat 5 a day of vegetables and fruits. And this is going to protect you all kinds of diseases like cancer and heart attacks and so on. This is what we're told all the time. And that's very nice if you really like eating vegetables all the time. But the question that most people wonder about is that if we want all those nutrients, why do we have to eat those vegetables? Why can't we just eat the pill and get the nutrients and then eat something more nice like cake instead? And in this lecture here, I'll try to talk about why carrots are quite important to eat, and similar things could be said about other vegetables, but I'm only talking about carrots today. And I'm still struggling a bit with this slide, but anyway, here we're getting. So what do we actually know, and how do we know that carrots, or if the carrots will protect against cancer? And there's a number of different kind of research we can do, and I'm just showing you one example here of a study which was done by one of my students here in Newcastle to find out if carrots actually do protect against cancer. So she had some mice which have a genetic mutation so that they naturally get tumors in the intestine. So they basically get colon cancer. Every single mouse gets several tumors on the average. And some of the mice, she would just give normal mouse food, which has all the nutrients that a mouse ever needs, which is quite important in this context. And the other half of the mice, they got mouse food, but where 20% of the feed was replaced with powder made out of just dried carrots. So they basically got their normal mouse food plus some carrot. And what you see on this slide here is that when we, at some point in the experiment, we killed the mice when the tumors were still small so that they wouldn't suffer. And we killed them, we opened and looked in their colon. What we found was that the mice which had been fed with the carrots, they only had approximately half as many tumors in their intestines as the carrots which had got the normal mouse food. And keep in mind that these mice were not deficient in nutrients, they got everything that a mouse needs that we know about from nutrition. So from this kind of experiment, it looks like there's something in carrots that actually protects against cancer. Of course, it doesn't tell us what it is, but it does say that there is an effect and that it's related specifically to carrots, because the carrot was the only difference between these mice. And you can see we both had some females and some males, and we saw the same effect in both groups. So does the same thing happen in humans? This is a bit more difficult because we can't control the diet of people, and we also, of course, most people fortunately don't get cancer ever. But there's quite a lot of studies like the one you see on this graph, where researchers have collected a lot of people, and they have asked them about what they eat in their diet. And then they, in this case here, they interviewed thousands and thousands of people. They were male smokers. The reason why they interviewed the male smokers is because we know that middle-aged male smokers have quite a high risk of getting lung cancer. And so in this way, they knew that they would get lots of data coming out of it. Because if you were interviewing 1,000 people randomly, then you might only have a handful of cancer cases. but in this group here, it's well known that they would have lots of cases. So they interviewed them about the diet, and they calculated then from the questionnaires, the data where people told them, what do we eat on a daily basis? Do we eat potatoes? Do we eat hamburgers? Do we have vegetables? And if so, which vegetables? The researchers then calculated in the diet how much there was of a compound called beta-carotene. And this compound, you can see the chemical structure on that on this figure. And this compound beta-carotene, it's the same as the orange color in carrots. So the reason why carrots are orange is because they got so much beta-carotene that it colors them. There's some carrots that are white because they don't have the beta-carotene, but most carrots are orange. What we could see in these data is that those people who have a lot of beta-carotene in their food, they have a much lower risk of cancer than the ones that have low intake of beta-carotene in their food. It's something like 20% less, which is a really big difference. If you eat one carrot, it typically contains about 6 milligrams of beta-carotene. And what you can see on this graph is that the group of smokers which had the highest content of beta-carotene in their diet, they had on the average 2.7 milligram per day, which sort of corresponds to about half a carrot per day. So those people who were eating half a carrot per day, out of 3,380, only about 85 of them got cancer. While this other group here, which were consuming less than 1 milligram, so that would be less than approximately 1 carrot per week. In this group, more than 100 people got cancer in this study. This study was done in Finland, and in this country, carrot is really the main source of beta-carotene in the food, so we can be quite certain that this group of people were eating many more carrots than this group of people. There's some countries where the diet is different, people don't eat a lot of carrots, and there the beta-carotene could come from other things. But we know in Finland these two things are very closely correlated. And I should also mention, I'm showing you this result from one study, but actually there are a lot of other similar studies, literally more than 100, and they all pretty much show the same tendency. So people who eat lots of carrots, they have less risk of beta-carotene. Sorry about that. So we now know that those people would have a high intake of beta-carotene. They're the same people who eat lots of carrots, and they're the ones who have a lower risk of cancer. So it would be reasonable to expect that this might, if the beta-carotene is beneficial and is protecting against cancer, then this could be the reason why it works. And so, because one thing is to know that carrots are eating carrots will protect you against cancer. That's very useful to know. But it would be even better if we could find out what is it in the carrot that is protecting us against cancer. In that case, certainly companies that are producing vitamin pills, they would very much like to know this so that they could make pills that will protect people against cancer and make lots of money that way. And it would also be a good way of making sure that people who don't like carrots, that they would also get the protection. So this study here continued to try to find out if the beta-carotene is in the carrot is that which is having the benefit. So by looking at these people here where you just look at the data from when they started, what they normally eat in their food when they started the study, and then nothing else happened to them. We know what happens if they don't get any extra beta-carotene, and we know that just a few carrots per week, these 3 or 4 carrots per week, will protect against cancer. But then what they did was that they took another group of people, the same number of people, they did the same thing with them, but they also gave them a supplement of 20 milligrams of beta-carotene per day, which corresponds to about 3 carrots per day. And these pills, so now they got something like 10 times higher amount of beta-carotene that they would get from the carrots. And of course, the researchers expected that they would have a lower risk of cancer, but what they found was that actually the risk is higher. And that was, of course, really disappointing for the researchers. They thought they were able to make a compound they could put in a pill and replace the carrot, but instead they were killing off some of their volunteers, which is not really nice when you're trying to save them. But now at least we know it's not the beta-carotene that's doing it. Now, some people say that this shows that actually carrots are not really protective. This doesn't work at all. And a lot of research about carrots basically stopped when this happened because it was quite a shock. But when you look very carefully at this graph, you can see that one thing is that those who get more beta-carotene, they have a higher risk of getting cancer. But the ones who eat the carrots in their normal diet and also get the supplement, they actually have a lower risk of cancer than those who don't eat carrots and get the supplement. So it looks like there's something else in the carrot, which is not beta-carotene and which can neutralize the effect of the beta-carotene to the effect that you can actually cope with getting too much beta-carotene as long as you just get this something else. And this something else is what we've then been working here in Newcastle to try to find out what that is. At the moment, we hypothesize that it's caused by this type of compound. Here you see an example of a chemical structure of a compound called falcarinol. These compounds are also present in the carrot. The dose is slightly less than for the beta-carotene, but not much. There's about 1 milligram per carrot of falcarinol. So when you're eating carrots, you get similar amounts of each of these with a factor of not more than 5 to 6. And we thought maybe this compound could have the protective effect of the carrot. So we've done some studies. Here you've seen one example with some cancer cells where we want to compare those different compounds. We want to compare the falcarinol and the beta-carotene. And so it was done using some cancer cells, and what the cancer cells were exposed to different concentrations of each of those compounds. And at low concentrations, 100% of the cells survived, so there was really no effect there. But when you get high concentrations of the falcarinol, then you can see that the survival goes down. Basically, this compound falcarinol kills the cancer cells, while if you take the beta-carotene in high concentrations, there's very little effect. I mean, you can get really, really high concentrations and nothing really happens. Also, nothing happens at low concentration. So the beta-carotene doesn't really affect the cells at all, while the falcarinol will kill cancer cells. At higher concentrations, it also kills cells which are not cancer cells. So you have to be a bit careful about that. But there is reason to believe that the falcarinol could have a beneficial effect in relation to cancer. Of course, cell cultures is one thing. It doesn't always work the same way in cell cultures as it does in humans. And so normally one will start first after you've discovered something might work. The next step then is to test it in animals. And what you see here are 2 data sets that I put together from 2 different studies. One study here is about beta-carotene, where they show that if they give rats, and these rats have been treated with a particular type of poison that gives the rats, makes the rat get cancer and get tumors. So in a way similar to the mouse model I showed you before. If they treat those mice with various concentrations of beta-carotene, and they've only tested the very high concentrations, they didn't test the low concentrations. What they saw was that at certain lower concentrations of beta-carotene, they did see a reduction of the number of tumors compared with the 100% that you see there. But as soon as the beta-carotene concentration was high, in the concentration in the food, the rat started getting more tumors than if they didn't have any beta-carotene. And here you see some data from a different trial, but with the same type of rats and the same type of tumors. And what they found here was that the falcarinol at the low doses, the sort of intermediate sized doses, reduced the number of tumors in the rats. They didn't use the high doses because that would have probably have killed the rats, because the problem with falcarinol is that it's actually quite toxic. But what you can see is that generally the falcarinol was more efficient at preventing tumors in the rat, and it works at much lower concentration than the beta-carotene. You need much more beta-carotene to get the same effect as you can get with falcarinol. So on this slide here, I'm sort of summarizing by comparing these two different compounds, and both of them, for both beta-carotene and falcarinol, most of that, or what we get of that compound in our diet overall, comes from carrots. So that's where we can say these are both carrot compounds. So beta-carotene is the type of compound called carotenoid, while falcarinol belongs to polyacetylenes. This is important because carrots also contain other carotenoids than beta-carotene, and it also contains other polyacetylenes than falcarinol. So there's actually additional amounts in the carrot that can have the same effect. About known benefits, we know that beta-carotene has a benefit in human nutrition because it can be converted into vitamin A. And so for people who are deficient in vitamin A, beta-carotene has a nutritional benefit. While for polyacetylenes, we don't know that there are any nutritional benefits, and there's never been tested in humans anywhere before. We know that animals can live happily for the whole life without ever eating any carrots. So polyacetylenes are probably not essential. Carotenoids have some effects on cells which are expected to be beneficial, antioxidant effect, and they can protect cells against UV damage, beta-carotene. Falcarinol has also been tested for cells. I showed you before that it can kill cancer cells at low doses. It also has an anti-inflammatory effect, so it reduces inflammation, which can be one of the precursors of cancer. If you have a high dose of beta-carotene, it increases your risk of cancer, but it's not actually toxic. You can eat food or pills with a high level of beta-carotene, and you may not get sick at all, but your risk of cancer goes up, like I showed you in that graph before. If you eat a high dose of falcarinol, it will damage your nervous system, and it can actually kill you. It is genuinely toxic. If you compare with caffeine, it's about twice as toxic in the sense that only half as many milligrams as caffeine is required to kill, for example, a mouse and probably also a human. Fortunately, there's no studies with humans, but it has been studied in mice. So compared with beta-carotene, a high dose of falcarinol is more dangerous because it can actually kill you in the short term, where beta-carotene in most cases will not kill you no matter how high the dose you get. There's sort of other effects of these two compounds. Carotenoids, if you eat a lot of carotenoids, a lot of beta-carotene, your skin goes orange, and some people think that looks nice and some don't like it. You might know some famous people with orange skin. If you get falcarinol on your skin, and that happens, for example, to people who work in factories where they are packaging carrots, or chefs who cut carrots every day, it can actually cause skin allergy. This is a well-known effect, which is definitely caused by the falcarinol, and that has been tested in humans. So we have some negative toxic effects of falcarinol. We have some nutritional benefits of beta-carotene. And there's also a big difference in how much research has been done about them. I did a very quick search and found out that there's at least 29,846 scientific papers which mention the word beta-carotene, while there's like that's more than 100 times as many as the papers about falcarinol. So one compound has been studied 100 times more than the other one. So if we don't know so much about this compound here, it's partially because it just hasn't been studied very much. And of course, one of the reasons why it hasn't been studied very much is because it's toxic and it causes skin allergy and it's not really nice to work with in an experimental context. So in conclusion, what we know about beta-carotene and what we can recommend in that context is that the beta-carotene is harmful in high doses, while the low doses, while what we can get if we eat carrots, and I said, say half a carrot a day, maybe 1 carrot a day, it'll give you some vitamin A and won't really do anything else. It won't harm you, it won't benefit you. So the conclusion about beta-carotene is eat carrots, but don't eat pills, because if you eat pills, you might get too much of it. I mean, not pills with beta-carotene. For falcarinol, we know that high doses are toxic. It can kill mice and also causes this allergy. Low doses probably protect against cancer based on the studies I showed you before. But we don't know, and it's not been tested in humans. So clearly the recommendation is exactly the same. Eat carrots, don't eat pills. Even if we do prove in a proper human study that eating falcarinol will protect against cancer, for most people, it's still not a good idea to use pills, because falcarinol is so toxic that if you were to have a jar with pills that you need to take every day, if you then ate 100 of them, you would be a very efficient way of committing suicide. So, but the thing about falcarinol, make sure that you don't get carrot juice on your skin for hours on a daily basis, because that can actually give you allergy. It doesn't give you allergy when you eat it, only when you get it on the skin. That's about half of the papers about falcarinol is about allergy. So we do know how it works in that context. So this was just to show you about how we do this research and what we found out until now, and why you should eat your carrots, but don't wait around for a pill that will replace the carrots, because that's probably not going to happen. If it does happen, it's probably still not a good idea to use it. So thank you very much for your attention, and I'll now like to see if there are any questions. And you can write in your chat if you have any questions, and there's one here already. Does it matter if the carrots are raw or cooked? That is a very good question. We have tried to find out. We have found out that cooking carrots generally does not destroy the falcarinol. If you make juice out of carrots, there are some enzymes in the carrots that seem to break down the falcarinol. So carrot juice might not be a good way of processing it. But if you cook carrots, like boiling them or steaming them, you still get the polyacetylines. Although if you cut them into little pieces and boil them and throw the boiling water away, you're going to lose not just the flavor of the carrots, but also the polyacetylines, because they basically disappear with the cooking water. We don't know about the bioavailability. If we know that for beta-carotene, it gets into your body much easier if you cook them than if they're raw. It doesn't look like that's that different with the falcarinol, but we don't know that yet. We haven't managed to make that experiment. We tried a couple of times, but it's really difficult to measure very small amounts of falcarinol. So we're still working on that problem. Yeah. Another question. Worth investigating level of toxicity for falcarinol for humans. It would be very worthwhile doing, yes, but the general view of ethics committees is that you need to prove the benefit before you expose your human volunteers to doses that might be toxic, which is very sensible. Because like the experiment I showed you before, where lots of people died because they took part in an experiment, because the experimenter didn't realize the toxicity was there. The ethics committees don't want us to repeat that, because that was really, really embarrassing for the scientists, and of course horrible in all different ways. So if we are going to use it as a treatment, we first need to prove that it works in humans at low doses. And if it works really well, we might then be able to, or be allowed to do trials to increase the dose and see if we see any toxicity. But we have to be very careful so we don't start doing long-term damage to the volunteers. And because the falcarinol affects the nervous system, it's actually quite dangerous. This is a very interesting question. If risk factors for lung cancer changes the effect of beta-carotene compared to non-smokers. It seems to be so that smokers, when they consume the same diet, they end up with less beta-carotene in their blood. And it's not quite understood how that is, but if there was an effect of this, then actually smoking would be protective, because it seems to reduce the ability of the beta-carotene to get into the blood. But of course, smoking by itself increases the risk of cancer. So that goes in both ways. However, what we've been investigating recently, haven't published it yet, is whether this correlation between carrot intake and cancer. And it seems to be very similar for a whole range of different types of cancer, not just lung cancer. And therefore, we don't think that smoking is actually doing anything in the context. It's just, the only point is that it's useful to do a study about cancer with smokers, because there you know that you will get quite a lot of cases within a short period of time. While if you want to start another type of cancer, it's difficult to find a population of people where you can predict that lots of these people will get a particular type of cancer within a short time. So I hope I answered that question as well as I can. So very interesting questions, and wish we had more answers, but there's definitely space for quite a lot of careers for people to investigate all these questions and other questions like that, because keep in mind, we still need 99 times as many papers before we get up on anything that just corresponds to what the research that has been done about beta-carotene, which definitely doesn't work in this context.